This lecture is the second of 5 lectures on the early season apple diseases. In the introductory lecture, you learned that early season apple diseases are defined based on the fact that they can start to develop as early as bud break, and primary infections can continue through petal fall. You also learned that intensive fungicide spray programs are used to simultaneously manage early season apple diseases, and that apple scab is the driver of these fungicide programs. In this lecture, we'll focus on apple scab, and the learning outcomes for this lecture are 1, to be able to describe the symptoms of apple scab, and 2, to describe the life cycle of apple scab and what role the sexual and asexual spores have in the disease cycle. Apple scab, which is the most common and recognizable apple disease worldwide, is caused by the Ascomycete Venturiae inequalis. The disease has a primary and secondary stage, which I will go into more detail on when we discuss the disease cycle. In poorly managed orchards, symptoms from primary infections can first appear on newly forming apples and prevent the apples from fully developing or cause misshapen fruit, as seen here in the first image. Primary infection on the leaves manifest as irregular shaped grayish-green spots. New infections have a velvety appearance as a result of asexual spore production. As the spots age on the leaves, they turn a dark brown color, and eventually the entire leaf turns yellow and drops from the tree. Scab lesions on fruit can range in size, but all of them are crusty and flaky, and depending on the variety, there may be a reddish color halo around the lesions, as shown in the third image here. You can see the reddish halo around a newly formed lesion. As the fruit mature, the spots will begin to coalesce and crack open, and eventually the apples drop to the ground. Obviously, these fruit are not suitable for the fresh market. However, they can be sold for cider or animal feed, but at reduced prices. If apple scab is well managed, there should be very few symptoms on the leaves or apples. The trees will have a full canopy, and there will be very little leaf or fruit drop. The apples will have no spots and will not be misshapen, as I'm showing here. So you can see a nice clean apple with no spots. In the larger image here of the trees, you can see that they have a full canopy and they have a lot of fruit production on them. If apple scab is not well managed, you will see the exact opposite, as shown in these 2 images. The fruit will be defoliated, the tree will be defoliated with little to no marketable fruit attached. There will be a lot of fruit drop, and the fruit will have multiple spots on them. Unlike the fungal fruit rots, which we will cover in future lectures, apple scab does not affect the flavor of the fruit. Unless there is extensive fruit cracking or secondary pathogens colonize the fruit can still be eaten. Crab apples are also susceptible to apple scab, and the symptoms are the same as those for fresh market apples. These are 2 apple trees in the Arboretum on the OSU Worcester campus. The tree in the forefront has obvious apple scab symptoms, but the tree behind is a cultivar that has resistance to apple scab. If we zoom in, we can see that the crabapple that is susceptible to scab has almost no fruit or leaves, but the tree with resistance has a lot of fruit and healthy leaves. So this bottom image is the tree that is susceptible to apple scab, and the top image is the tree that has resistance to apple scab. Crab apples are often planted in commercial orchards as they are excellent pollinator attractors. Because they are used to attract pollinators, it is important that growers plant resistant cultivars, otherwise the trees can be a source of inoculum. In order to effectively manage apple scab, a good understanding of the disease cycle is required. Apple scab is a polycyclic disease, which means that the fungus produces more than one infection per crop cycle. In contrast, monocyclic diseases or pathogens produce only one infection per cycle. The primary inoculum produced by Venturae inoculis are the sexual spores or ascospores, which are produced within pseudothecia. So here's the primary infection cycle and the secondary infection cycle, and this is what makes apple scab a polycyclic disease. Over here you can see an image of a pseudothecium, and within the pseudothecium are the ascospores. It's this primary inoculum that causes infections, and then large number of asexual conidiospores are produced at each infection site caused by the primary inoculum, and these spores can cause new secondary infections that produce more asexual spores and more infections, and the cycle continues. The pathogen overwinters in fallen leaf and fruit debris, and it's overwintering as mycelium in immature pseudothecia, or in this case, one is a pseudothecium. So pseudothecia are cavities located within a dense mat of fungal mycelia called stroma, as shown here. The asci contain 8 ascospores that develop within the pseudothecia and are released in the spring. So in the first image here we see immature pseudothecium. So you can see the stroma along the bottom, and you can see the asci. So here's one ascum, and then there's multiple asci in here. On the right is a mature pseudothecium. Again, here's the stroma along the bottom, and now you can see that within the mature pseudothecium, the ascospores have developed. And again, these ascospores develop in the early spring. So primary infections are the infections that can cause the most damage and are thus the focus of a management program. If primary infections are controlled, the number of secondary infections will be significantly reduced. Primary infections are initiated in the spring upon the release of ascospores, and this occurs at bud break or close to bud break. Depending on the temperature and the amount of rainfall, spores can be released through the end of bloom, which is typically the end of June in Ohio. So the ascum is what we call bitunicate, meaning that they have a double cell wall. So you can see that double cell wall here on the ascum. This structure is important for the release of the ascospores in the spring. Each ascus contains 8 ascospores that are paired in a linear arrangement. So you can see that they're paired here in a linear arrangement. The ascospores are brown. They're 2-celled spores, and they have a characteristic footprint shape, as you can see here, or a boot shape. The shape of ascospores inspired the Latin name for apple scab, inequalis, which refers to the unequal size of the cells. At maturity, both layers of the ascum rupture, so both of these layers rupture at the tip, and the endoascus takes up water and extends out of the pseudothecium, resulting in the release of spores. And the release of spores can occur over the entire primary infection cycle, so from bud break to petal drop. There is only one cycle of ascospore production, however, and one infection per season. It is a diurnal pattern of release, meaning that it occurs every day and every night as long as conditions are favorable and until all the spores have been released. As I mentioned again, this occurs from bud break until the end of bloom. Wet and cool conditions favor the release of ascospores, and the spores are carried to blossoms and leaves by rain and wind. Very little rain, about 1/10 of an inch, is needed to initiate extension of the endoascus and spore discharge. Spores are generally released after a nocturnal rain during the day, and so the release of the ascospores is light dependent. Less than 20% of the ascospores will be released during darkness. The top image here shows pseudothecia on leaf debris before a rain event, and then on the bottom after the rain event. And you can see that the pseudothecia after a rain event are globular compared to those before the rain event. And this globular structure then is the asci with the ascospores. Secondary apple scab is initiated by conidia on the fruit and leaf lesions that developed from the initial primary infections by the ascospores. So this image here, you can see the grayish brown color. These are the conidia. And you can also see it on the fruit in the center, this brownish gray color. It's actually a little bit easier to see up at the top. It looks more like dirt or a smudge within the center of the lesion. So conidia are produced in large amounts, as you can see, and splashed to new infection sites throughout the growing season. The number of secondary infections is directly related to the number of primary infections that occurred. If the primary infections are controlled, secondary scab has little impact on crop quality and yield. This shows a close-up of the secondary leaf lesion that you just saw, and you can see the conidiophores and conidia of the fungus. So here's a conidiophore and then the conidia coming off. Management of apple scab focuses heavily on reducing primary inoculum in the orchard and preventing primary infections from occurring. As I just mentioned, if primary apple scab infections are controlled, secondary infections will be minimal and have little impact on crop quality or yield. In the introductory lecture, we discussed the use of host resistance to control early season apple diseases, including apple scab. Developing varieties with apple scab resistance has been a high priority for apple breeders, so there are many varieties that are moderately to highly resistant to the apple scab fungus. In the Midwest and northeastern United States, apple scab pressure is high, as average temperatures in this region of the US favor disease development. For this reason, we always recommend that homeowners specifically select varieties with resistance to apple scab. For commercial growers, they have a bit more flexibility because they can implement an apple scab fungicide spray program. Because the fungicide spray program needs to happen on a weekly basis, so it's an intensive program, homeowners typically will not be applying fungicides to that degree. So in the next lecture, we will discuss apple scab management, focusing on primary scab management. For primary scab management, forecasting models, fungicides, and best cultural and sanitation practices can be integrated to effectively and consistently control primary infections. We will focus on these 3 strategies in our next lecture.